I have the joy of introducing our fearless leader, Dr. Brad Dickerson. Dr. Brad Dickerson is the director of the MGH Frontotemporal Disorders Unit and the Neuroimaging Lab in Boston. He is a staff behavioral neurologist in the MGH Memory Disorders Unit and co-investigator on the neuroimaging core of the Alzheimer's Disease Research Center. He's also a professor of neurology at Harvard Medical School. He is a husband, a dad, and a friend to many of us here. Brad makes work fun, even on the tough days, and he is always there to show us that he cares. So, let's show Brad how much we care and give him a round of applause.
Thank you, Katie. It's always so moving to hear you speak on these nights. You're just so good at it. I don't know how you do it, but thank you. So in 2007, my good friend and colleague Daisy Hochberg and I started the MGH FTD Unit. I think she would answer the phone sometimes, I would take the mail out sometimes. We did pretty much everything. Obviously we had some great staff that we were affiliated with who helped, but it was really just the two of us in the early days. Thank you, Daisy, for sticking with our labor of love and watching it grow into the picture from the Gala last year that you see here. Thanks also to many mentors and colleagues who provided a lot of support and referrals and other activities along the way. A special shout out to Bruce Rosen and the Martino Center family for doing the amazing work that you do and allowing us to stay ahead of the cutting edge with our scanning imaging techniques by working with you guys so closely over the years. I'd also like to thank the amazing team of people who are the FTD unit. You saw us up here getting our photo. This is a group of people whose incredibly high level of competency, compassion, and commitment keep our operations running and keep us growing. You keep seeing the picture getting bigger every year because through all the hard work, we keep expanding and starting new initiatives. I'd also like to thank our talented featured artists who generously shared their beautiful work with us in the reception and art show before we came in here. Thank you very much. That's one of many things that I think makes this night so unique and really powerful. Also, you, our community. You did not choose this community, whether you're a person living with one of these conditions, a family member, or a loved one, but I'd like to say it's better for what you bring to it. Thank you. We would not be here tonight if it weren't for Sybil and Kimon Sermos. I vividly remember sitting in clinic when Sybil said, you know, being professional artists, this is what we do, and we'd love to do something like an art show for you guys in support of the program. And Katie and I brainstormed with them for a while and basically came up with the idea for the first night for the arts for the FTD unit. So thanks to Sybil and Kimon, and we miss you, and your legacy is living on through this event. So thank you to Sybil and Kimon's family. And special thanks to Katie Brandt and to Amy Almeida. Really the partnerships that we have The partnerships that we have enjoyed developing over the years have led to many great ideas, and I think this is one of them, and it keeps going. It keeps growing, it keeps expanding, new things happen, And we wouldn't be here without you guys doing what you do. So thank you. I want to just take a few minutes to review with you some of what we do in our day-to-day work. And I always like to highlight a few bits of progress from research that are things that are new that I wasn't able to say last year because we didn't have them yet. So we do a lot of work on care and support today, but research for the cure and better treatments is really what we are here for tonight. And we've been able to leverage a lot of our initial pilot experience with what's called digital voice biomarkers, which I'll talk briefly more about, and with cluster scanning MRI techniques. And these were all things that were supported by seed funds that came from nights like tonight when people like you made generous donations to get us started with some new ideas. And these projects really wouldn't be funded by an organization because the ideas are too new. We don't have any preliminary data as we are asked for to show that it might work. And so the philanthropic funds that you're donating tonight go directly to the support of new ideas that ultimately we hope to leverage into foundation or NIH or industry-sponsored grants, which we've been able to do with multiple examples that I'll highlight briefly tonight. So, when you want to know what does this money go to, we put it directly to the people and the projects that are advancing our science. One of the studies that we've talked about for the last few galas was a study led by Katie Brandt. Yes, she's also a researcher, believe it or not. She's amazing at it. And Erin Cron, who's our project manager, and the two of them, with a lot of other members of our team, developed this study that we ultimately got funded by the National Institute on Aging of an educational intervention for caregivers, thinking about advanced care planning, meaning planning for down the road from where things are right now. And it's complete. We enrolled over 130 caregivers. It was a clinical trial comparing two different types of education interventions with people, and it turns out it doesn't really matter so much how you do it, it's that you do it. And we now have evidence to support that, which is what the federal government wants to see in order to pay for these kinds of things, which we know tend to get done outside of what is paid for as part of a regular medical care visit. So hopefully we'll be able to see impact at a variety of levels from this kind of research. So congratulations to Katie, Erin, and other members of our team for getting this done. So now I'm going to go around the circle. This is Lauren's favorite time of the night. And just talk with the patient at the center. We always talk about patient-centered research, person-centered research, about the way that we go from the measurement of people's symptoms to the development of new treatments. And so first we have to have scales to measure what kinds of symptoms a person is having, what is difficult for them, and whether it's getting worse, whether it's staying the same, whether it might be getting better. And so it's a little bit embarrassing, I have to say, in the 21st century to realize, even though Dr. Eldife looks great doing the neurologic exam, it's a little embarrassing to recognize that we're using methods to rate people's symptoms or grade people's symptoms that were developed in the 1980s or 1990s. They're really, should be outdated at this point, but this is the cutting edge of what we use. And so I think we all recognize the need to improve those methods. And so there's a colleague of ours who's built the NeuroBooth, which is something that Dr. Wong on our team has been working with our colleague to bring people in to use sensors, various types of sensors that we all interact with every day on our phones or with our computers or other kinds of sensors, accelerometers on our phones that measure movement, so that the neurologic exam can no longer be simply rated by an expert neurologist or neuropsychiatrist, which is important, but so it also can be supported by quantitative information that comes from all of the amazing sensor technology that are embedded in our day-to-day lives and now are embedded in our clinic. And we've got a progressive aphasia severity scale that that Daisy and I and other colleagues developed in the early days of the FTD unit, when we realized patients with progressive aphasia don't show up as having abnormalities on the typical grading scales that are used in work on with patients with Alzheimer's disease or frontotemporal dementia, because there's not a scale like this that allows us to measure these symptoms. Kind of a simple idea, but it was groundbreaking enough that you can see the map of people all around the world that continue every week to request copies of the scale, and we have a clinical trial that is requesting that they use the scale now. And again, this is a scale that's rated by expert clinicians that are trained in these topics, which is important, but Dr. Rezai and Dr. Wong in our group are also using digital biomarkers, which are basically what you get when you look at things that happen on your phone, when you talk to Siri, when you talk to Alexa. So there's tremendous amount of information in my voice right now, the words that I'm using, the pauses that I inadvertently make from time to time when I'm trying to think of what that exact word is that doesn't quite come to me, or the thought. And so when you use computational methods, computer methods, to measure the acoustic properties or the linguistic properties of what people say, there's a tremendous amount of information in it, and it turns out that it can tell us if a person has cognitive impairment and potentially what type of cognitive impairment they may have. And that ultimately is being validated by things like neuropsychological assessments and traditional tools, but it may be the wave of the future where we ask people to give standard recordings of responses to questions, maybe even before they come into clinic, and then we can have a sense of what kinds of issues are happening with them before we even start to do our formal examinations. And those are the kinds of things that I think are more objective measures that we can use in our clinical trials of new treatments to try to see if these types of interventions are able to either stabilize people's condition, slow it down, or possibly even improve it. So really cutting-edge outcome measures. Then we've got imaging that measures changes in the structure and the function of the brain. And so one of the amazing things about this specific condition, one of the conditions we work on called primary progressive aphasia, is that at the beginning, it really just affects a person's language. And if you look on the top image here, those are the parts of the brain that are normally active when you're doing what I'm doing right now or what you're doing right now, which is either producing hopefully coherent speech or hopefully not falling asleep and comprehending what people are saying to you. That's the normal language network. And if you look at the areas of the brain that are shrinking when a person has primary progressive aphasia, that's what you see on the bottom. So those are areas that are getting smaller and not doing their job, and it's very specific to the language network, at least at the beginning. So we don't quite know how that works, but members of our team, Dr. Turotoglu and Dr. Eldaif, are using transcranial magnetic stimulation to try to see if we can improve the functioning of this network, as even though we're fighting an uphill battle, but what we ultimately want to be able to do is hopefully slow down the progression, but also improve the functioning of those networks in the brain so that maybe people's symptoms get better. And I'm really proud to say that their first report of a patient that did show some improvement, both in the brain function and in language performance, just came out today. So congratulations on the new publication. We've got a variety of molecules that are at the fundamental root of the problem in these conditions. You've probably heard about amyloid plaques, tau tangles. These are what disrupt the function of the brain circuits in the brain when the circuit is not working properly. So we can image tau, we can image amyloid. We don't yet have the ability to image certain types of the proteins that are key to frontotemporal lobar degeneration, but we're quite successful at being able to image these in Alzheimer's disease. And we now have spinal fluid and even blood measures that can reflect some of these molecular abnormalities as well. And so what we know with those molecular biomarkers is we can see the progression of the problem over time. What's really amazing, and this is the first time I can say this to you in tonight's event, is that because the first so-called disease-modifying therapy for Alzheimer's disease was approved last year, and we started using it, just not that long ago, really in early 2024. Normally, the amyloid in Alzheimer's disease gradually increases over time. That's what the graph shows here with the black line showing a gradual increase of the problem, which is the amyloid protein. But when people are given this new drug that is an antibody against amyloid that tries to get it out of the brain, you can see on the right graph that this is a 63-year-old woman with early-stage Alzheimer's disease who started receiving lecanemab, and her amyloid levels decreased substantially, much more so than what would ever be possible without a treatment like this. So I think we're really now, for the first time, able to say that we've got a disease-modifying therapy for one of these neurodegenerative diseases that leads to dementia. And I think we're not that far from having others as well. So this is a real milestone. Now, we have to come up with ideas for how to develop new drugs or screen existing drugs to see if they might work in models of these diseases. A lot of times they're animal models. Sometimes they're models in cell culture. One of the things some collaborators that we work with at Mass General do is they reprogram skin samples that I get from a little skin biopsy from people into stem cells, and then those cells can be turned to any kind of cell in the body, but they have that specific person's genetic makeup. And so if a person has a particular genetic abnormality, for example, that leads to tau protein accumulation in FTD, you can measure things that are going wrong in the cell dish in that human cell-derived laboratory model of the disease. And so what that means is you can screen treatments. So on the left is an image of the brain cells in the dish that are grown in this way that are labeled for the bad type of tau protein in yellow. And you can see a lot of that all over the screenshot. And then on the right, there's a drug that was screened to see if it might be able to reverse or slow down some of those changes, and it really gets rid of most of the yellow. So these are some of the kinds of laboratory methods that are being used to screen either existing drugs that we think could be repurposed to treat these diseases, or new drugs that are being developed to try to see if they might be possible to treat some of these diseases. So then, what about clinical trials? We have to bring it back to people and try the drugs in people or other interventions and see if they might help. And so there's a specific type of FTD that's related to a genetic abnormality in a gene called progranulin. And if you don't have that normal functioning gene, your progranulin levels are too low. That's what's shown on the left of the graph here. So the people in the red circle are people with this genetic abnormality, and their levels of this protein are too low. And there are a lot of important functions that that protein does. It's not clear why people can live for 50 years before they have symptoms, but that's what happens. They live into their 50s and then will develop symptoms, or on average in their 50s, and have FTD that progresses. And so the question is, could we simply restore that deficit and possibly cure the disease? And so there's been a number of trials. There are multiple trials going on right now in patients with this condition to try to see if there's preliminary evidence that it might work. And this was the first trial that was published on this topic ever, just this year. It's a gene therapy treatment where essentially the gene was replaced. It was a very technically complicated approach, but it appears safe. You know, all the discussions about gene therapy, is it safe? Is it going to cause other kinds of problems? It appears to be safe, at least preliminarily, and it corrects several of the biological problems that we know are at work in the cells when people have this problem. So what you can see in the graphs at the bottom is just that when the patients were first screened at baseline, they had abnormally high levels of these proteins. And as they were followed over time, after they got the gene therapy, those protein levels started to normalize. We don't know if it's going to help their symptoms. We don't know if it's going to ultimately be a cure. But this was the first time ever that a gene therapy for a type of FTD was tested and looks, at least at the beginning, like it might be A, hitting the target, it might be doing what it's supposed to be doing, and B, might be rescuing some of the biological problems that we think are going on in the brain that lead to the degeneration. So it's yet another very significant milestone that I think we have to acknowledge as another major milestone of 2024. I'll just close in the last few slides by just mentioning many of you are part of the All-FTD study. This is a major consortium study that is at all these centers around the country. There's a similar one in Europe, and together we work on an initiative called the FTD Prevention Initiative, and that's really a global effort across many countries to try to get to a point where we can either significantly help people with treatments or possibly even find ways to prevent FTD in people with genetic forms of it that you could give treatment to before they have symptoms. And then we've also got the LEADS study, Longitudinal Early-Onset AD study, that I'm proud to be one of the leaders of, that has sites all around the country, and a number of you are part of that study. This is focused on young-onset Alzheimer's disease, and we've been successful at enrolling 400 people with early-onset Alzheimer's disease younger than age 65, which is the largest sample of people ever studied in a natural history study, and we're encouraging people to stay in the study if they get on the new treatment so that we'll be able to really see those levels come down like I showed you before. So these are two major consortia that encompass most of the kind of work that we do in the FTD unit, and we're part of those and very happy to be doing the work we do in partnership with many wonderful colleagues like Katie mentioned in her talk. So all of the research to work toward a cure for these conditions depends on participation by you and other people like you. We cannot do it without your participation in any way that works for you. And together, I really do believe that we're going to win the fight against dementia.